Okay. Are there any questions about your lab kit? Everybody good with your lab kit? We'll finish going over it, Nate, at the end. We're going to start up our regular quizzes again next week. So you're going to have a quiz on mitosis and meiosis when you come into lab next week. And I'm really glad I got through what I did in lecture. The timing was pretty good, but we're going to start on page 55 and we're going to go through first the cell cycle, and then we're going to go through mitosis and meiosis. Now, my suggestion to you is, along with your lab book, get some pieces of scrap paper. Okay, so I lectured basically for 2 lectures on mitosis and meiosis, and I find it works a lot better for you if you have some skin in the game. So drawing some things out for the purposes of remembering what the different things are. So I would encourage you to make sure you're doing that. The other thing that I'm going to do is I'm going to do a lot of drawing here, and I will ask you questions as I go along. I will try to make it as interactive as possible. This is a very hard one to be that interactive. If we were in class, it works great because you would all get what I have, which is this handy dandy little set of beads, and you'd get to move the beads around while I was moving the beads around. And instead, I'm going to have to do that, and you're going to have to be following me. So a couple tricks for this. One is write things, draw along with me, whatever helps you remember. Also, this is a very long lab. If you look at how far it goes from page 55 to 72. But on page 72, there are microscopic observations. Now, we are not going to do, we're going to use the virtual microscope, and that's going to be your photo assignment. So you're going to upload the stages from the virtual microscope. So, and that link is in the mitosis lab. But this last page, you don't have to hand anything to me. So I would take that opportunity to draw in these different phases when we do them. Okay, so that's a good place to draw. And obviously you've got scrap paper, but this is a lot of information. Almost all of it you've heard now. So this is the second time through, and I would anticipate it should be solidifying it, not totally confusing you. So you need to stop me if there's things that you're not getting, because this is really the last time we're going to go through it. Okay, so keep that in mind. All right, page 55, and I'm going to switch here because I'm going to try to do a lot of this on my paper. So I'm starting with a big piece of paper, and let's start with what is that? If you had to guess, what is that?
It's a chromosome.
It is a chromosome. Thank you, Taylor. That is a chromosome. And I'm a terrible drawer, so you're going to have to bear with me here. What is that?
A pair of chromosomes.
Is it a pair of chromosomes? Still a chromosome. What is it actually, though? So this is one chromosome. This is also one chromosome, right? Remember that. And that's one of the hardest things when we go through, is to remember that this is chromosome 4. and this is chromosome 4. The difference is these two things right here. Those are called sister chromatids. Okay, those are sister chromatids. And I know, I apologize, I'm writing sideways, but. So these are called sister chromatids, and they are identical to one another. Let me try this. Whoops, I did not do that well, but what do we call this? Besides a circle, I know, but when it comes to a cell, what would this represent? I'll give you a hint.
Is that interphase?
It is interphase, exactly. So this was gap 1, right? G1. And then what one comes next? Anybody remember?
S.
S. And then what comes after that?
G2.
G2. And then after that, this is mitosis. And my first stage is going to be prophase and metaphase and anaphase and telophase. And then, whoops, the next, cytokinesis. Right? So that should look familiar. What's happening in G1? Anybody remember what happens in G1? Let's think about it for a second. If this is my cell cycle and cytokinesis is at the end, if I start with one cell and I go around the cell cycle, how many cells do I have after cytokinesis? I start with one cell here and I go around this way. And cytokinesis, remember, is where my cells separate. So how many do I have at the end of my cell cycle when after cytokinesis?
2.
2. All right. So I start with one cell and I go to 2. This process then has to prepare, really up to the end of G2, this process has to prepare my cell to go from one cell to 2 cells. So G1 is going to be to make organelles. I need more organelles, so I'm going to make organelles here. Anybody remember what happens here in S phase? I'll give you a hint. It stands for synthesis. You remember what S phase is, Tyler?
I know the name. I just don't know what it does.
So this is S phase, and S phase is when our one chromosome, right, goes into sister chromatids. It replicates itself exactly. So we're going from our chromosome looking like this in G1 to our chromosome looking like this during S phase. So that's replication of our chromosome. Now we have 2 copies, even though we only call this a single chromosome. And that's because it still is. This is still chromosome 4, whether it's got 2 little wings on it or 1. Okay, so now I've got my chromosomes looking like this. What happens in G2? We're doing all the rest of the stuff, right? We're doing the rest of the stuff that gets our cell ready. We may need some more proteins, right? We know we have to make a lot of proteins in our cell. So maybe we're making proteins and maybe some extra phospholipids so we can make our bilayer, right? We may make some glycolipids that are going to tell every other cell in the body what type of cell that is. Okay, so that's doing all the rest of the stuff that gets our cell ready to divide. And somebody told me what this whole stage was. We lump it all together, and what did we call this whole part?
Interphase.
Interphase. All right. Perfect. So there's our cell cycle. And if you're looking at the top of page 56, there's your cell cycle. And you know now that G1 is where the organelles are going to be made. S phase is where we're going to replicate our DNA. So we're going to go from this being our chromosome to our chromosome looking like this and having twice as much material. Identical material, but twice as much. And we know that G2 is going to make other things. There's going to be some protein synthesis. That's probably what's going to happen most, is protein synthesis. And then our last stage, which we lump together as mitosis, is going to be our last stage. And that's when our cell is actually going to distribute the chromosomes and divide. So in mitosis, we're going to distribute the chromosomes, and then we're going to divide into two cells. Okay. So right now we're only talking about mitosis. Okay. So remember that mitosis is when I cut myself, I cut my hand, or I cut my finger. And I need to fill that void, that cut, with new cells. So I'm going to have cells that are next to that space that are going to divide to make new cells to fill that space. So we'll start with one parent cell and we'll go to two daughter cells. But I want those daughter cells to still be skin cells. I want them to do the exact same function as the other cell, right? As the parent. So when I get done, I'm going to have two cells, two daughter cells, that are genetically identical to the parent cell. Okay, so mitosis, remember, is going to be two daughter cells, and they're going to be genetically identical. Genetically identical to the parent and to each other, right? Now, the last term that we want to be reminded of here again is the term diploid. Diploid. And I was trying to think of how to do this, and I didn't have time before lab, but let's just say, let me switch to this for a second. And I have showed you a karyotype, and you'll recognize this when I go through it, but it has that chromosomes 1, 2, 3, 4, 5. Okay, it had chromosomes all the way down to 22, and then it had an X and a Y. Do you remember what I'm talking about? And it showed that we had a chromosome from mom and a chromosome from dad, and we had a second chromosome, chromosome number 2, one from mom and one from dad. And it basically looked like that, right? All the way through. They were about the same size. They had darker portions and lighter portions. So this is a diploid cell. There are 2 copies of each chromosome. That's called diploid. So diploid. What happens if this was my chromosome 1, and this was my chromosome 2, 3, 4, and 5, etc.? This is haploid. Hap and half. Hap, H-A-P, is the word, but think of it as half of the number. So haploid equals half. But what I want to point out, and what I think people get messed up with, is this haploid set of chromosomes is still a complete set of chromosomes. Haploid means that from 1 up to 23, we've got a chromosome for each 1 through 23. They have different things on them. Diploid means you have 2 copies of chromosome 1, 2 copies of chromosome 2, of chromosome 3, all the way up to 23. For some reason, people have a hard time with that, because we'll say our sperm and our egg, when we get through meiosis, are going to have 23 chromosomes. But that is a haploid set, meaning it's still going to have all of the chromosomes. It's just only going to have 1 of each, versus diploid, where there'll be 2 of each. Okay, so those are the other word we have to put in our memory banks for talking about mitosis. All right, so when we get done, we're going to go from a diploid cell to 2 daughter cells that are genetically identical, and they are going to be diploid cells. So if we're talking about humans, that's going to be 2n, and n is going to be the number of chromosomes. So diploid is equal to 2n. So for humans, it's going to be 2 times 23, or 46. Okay? Any questions with that? That's going to be important. All of this stuff that we've kind of used to set things up is going to help when we look at the steps of mitosis again. So if you have questions, this is the time to ask. Whoop, hold on. Sorry, Tyler, here I saw you put an answer and I didn't have the chat up, so. Okay. I have the chat up now. Okay, so that gets us started. Let's take a look at what's going to happen after interphase and going through mitosis. So let me move this out of the way. We may want to come back to it. I'm not quite sure where it's going to go. Well, try without that. I was hoping, because this is tile, I was thinking maybe I'd end up with little bumpy writing, but so far so good. Well, so I have my paper here, and my paper is going to be a cell. Now it's going to have a cell written on it. I shouldn't say it's going to be a cell. But let's start with my cell. So it's been through, that's a pretty pathetic looking cell, but I'm actually going to make that the nucleus. So I have my cell here, the bigger one, and then this is going to be my nucleus. It's lucky I was a Girl Scout leader. I have tons of, well, I have one roll of paper left. I have these big rolls of paper that I got at a factory. All right. So I'm going to put all my, these are going to be my chromosomes. And they're all going to be in the nucleus. And they're all going to be kind of mixed up in the nucleus, because remember we talked about, first of all, the nucleus is going to house the genetic material. So my DNA is going to be in the nucleus. So here's my pretty DNA in the nucleus. Now, in interphase, right, I'm now going through gap 1, and my cell is making more organelles, and then I'm going to S phase. And I probably should have started this a little differently. Hold on. Okay. So here is my cell. And I get to S phase, and I'm going to duplicate my chromosomes. Okay, so I started out with this chromosome 4, and I duplicated it. So now I have chromosome 4, but in the form of sister chromatids. I'm duplicating my dad's chromosome 4. And let's see, I'm going to duplicate chromosome 22 and mom's chromosome 22. And while I've done that, all of a sudden, now remember, I'm going to have 23 sets of chromosomes. Or actually, I'm going to have 46 total chromosomes, 2 sets of 23. So I'm going to get this down to a workable number now. And I've now replicated my chromosomes. Going into prophase, that's my first phase of mitosis, my chromosomes are going to condense. And every time I go to prophase, write this somewhere. Every time you go to prophase, let me see. I think it might be, talks about interphase. Hold on. Go to the bottom of page 59. And you'll see that it talks about prophase. And somewhere there, I want you to put the 3 things that happen in prophase, any prophase, okay? The 3 things that are going to happen in prophase are my chromosomes are going to condense. That means they're going to become visible. Instead of looking like a ball of yarn, I'm going to be able to visualize them. So I'll write that down. Somewhere I lost my. I had a really good marker a second ago. I don't know where that disappeared to. Okay, but my chromosomes are going to condense. That means they're going to be made visible. So there's my first thing. My second thing, and it doesn't matter which order, all of these are going to happen during prophase. Prophase. The second thing is my nuclear membrane is going to start to fragment. It's going to break up. Breaks. All right. And the third thing, does anyone remember the third thing? I'll give you a hint. What's this? Anybody remember what those are?
The spindles?
They are going to make the spindle. Absolutely. Those are called centrioles. And those are what the spindle is going to be made from. So good answer. All right. And the centrioles are going to migrate to the poles. So one is going to end up on one side and one is going to end up at the opposite pole. Every prophase is going to have that happen, whether it be prophase of mitosis, prophase 1 of meiosis, or prophase 2 of meiosis. Okay, everybody with me? So let's move on. And now I don't have a nuclear membrane anymore because that's all fragmented. That means this entire cell is free for me to undergo mitosis. Remember, I have my centrioles at the opposite poles, which are going to make my spindle. And remember, my spindle is going to be fibers, microtubules that come down and they're going to grab on to, I lost my hairs here. It's going to grab on to one side of each of my pairs. Okay, so this fiber right here is going to grab this sister chromatid, and it's going to grab at the centromere, this part right here, the centromere. So it's going to grab at the centromere. This side over here is going to grab at this other centromere. And that's going to happen. And the result is going to be that during metaphase, all of my chromosomes are going to be lined up at the center. So I'm going to have this row of chromosomes all lined up. That's metaphase. Meta meaning middle. my next phase, what is my next phase? Anybody remember what my next phase is called?
Anaphase?
Anaphase. All right. So anybody remember what happens during anaphase? Now that I have these fibers, my spindle fibers attached to my centromere, I'm going to move these out of the way for just a second. But each of my spindle fibers is going to contract, meaning it's going to get smaller, and it's going to move this direction on this side, and it's going to move to this on this direction and this side. It's going to take with it the chromosome that it's attached to. Now, all of a sudden, we've separated those sister chromatids. So this one is going to head this way, and this one is going to head this way. And that's going to happen to each of our sets. Yep. Okay. And as soon as those start to separate from one another, that's called anaphase. There is anaphase. So these are going to pull apart, and they are going to pull up toward our centrioles. This actually is now called a centrosome, but don't worry about that. So all of these are going to pull up into the top of this spindle. And remember, this is our whole cell now. So once they get up near the top, all of these are going to decondense again. Okay, now they're all going to decondense. And when they decondense, all right, let's make a list for telophase. What we expect to see for telophase. So it goes prophase, metaphase, anaphase is when they're starting to separate. Telophase is going to be marked by several things. One is that the chromosomes are going to decondense. So we've got decondensing chromosomes. Secondly, every cell needs a nucleus, right? So we've got to make a new nucleus. So we're going to start seeing a nuclear membrane form around our chromosomes. So we're going to have new nuclear membrane. And the other thing that we're going to see, and it's hard for me to write it on here. Let me see if I can maybe make this paper a little wider here. What was that called? Anybody remember what this is called? No, it's not a butt. Nobody remembers what that's called? That's called cleavage. You knew what it was. You just didn't want to say it. Okay, so that's cleavage, is where we start to see the original cell membrane start to indent. Why is it going to indent? Because it's going to separate into two cells. So telophase is going to have the decondensing chromosomes, the new nuclear membrane formation, and cleavage. Okay. The last stage, I've already used this term a couple times. Does anybody remember when we completely separate these into two cells? What is that called? Cytokinesis, right, Tyler? So that's our last step, is cytokinesis. So when this separates the two cells, that's cytokinesis. Cytokinesis. All right, so look back at your book on page 58, and you can follow right along 58, and you can see what I did. You can also look above on the pictures, and on those pictures, those are actual micrographs of a cell undergoing mitosis. So you're seeing the various stages, you're seeing the decondensing of the chromosomes. Well, you aren't actually at the end very well, but you are a little bit. But you can, that would be what you would see. So when you go to look at the virtual microscope, you'll see variations of each of these. All right. So, and now you know why. What you're actually seeing is those chromosomes. Is there any questions about mitosis? So you could all do this, or you could all recognize a cell that is doing this and what phase it's in.
So the last phase, you wrote the word cytokinesis.
Yep.
That's when that dotted line that connects the two cleavages, is that the splitting, or is that just
It is. It's the process of splitting the cells into two cells. So they separate the cytoplasm and they build more cell wall, or not cell wall, sorry, cell membrane along the middle of it. And that makes it two complete cells. So the next picture would be, what? The next picture would be two individual cells. I hate to waste my paper. I might have to grab some more, but my next one would be this. Skip the cleavage here, right? But I would have two new cells. These would be my nucleus. So that would follow this.
Got it.
Okay. All right. So down at the bottom of page 60, it says the summary of mitotic cell division. And it says the nuclei in the daughter cells have blank number of chromosomes as the parent cell had. Same, right, Tyler? And mitosis is cell division in which the chromosome number, does it change or stay the same? Our chromosome number going to change, or is it going to stay the same? Megan?
It stays the same, right?
That's right. It's going to remain constant. Yep. All right. Now, what I'm going to do, I think that might be the easiest way to do this, is I'm going to go through, just like I did. Let me grab another couple pieces of paper. I have more, but I don't think I have enough. I was cutting pieces of paper in my meeting. Okay. So let me go back here. Get rid of some of these. I do not know what happened to my good marker. It was so much nicer to use. This one's so heavy. I couldn't have gone far. I haven't gone anywhere. I don't know. Go figure. Oh, there it is. Okay. All right. So now we're going to do meiosis. So the first thing I want you to do, going down on page 61, find where it says prophase 1. And write crossing over. That should sound familiar to you because we talked about crossing over in class. It was one of our mechanisms of genetic variability. Actually, Tyler, I figured you fell asleep. That was why you hadn't answered me. All right. And under metaphase 1, or next to metaphase 1, I want you to write independent assortment. And next to that word, write random alignment. Independent assortment and random alignment are the same thing. You may hear it referred to either way. Okay. So this time, start, actually, you know what I'll do? I'll start right on this piece of paper because prophase is going to start the same. So let me get all my stuff here. Let me pull this and this, that. Okay. So I'm back to my cell and all of my chromosomes are in my cell and they're in the form of chromatin. That's kind of the yarn-like. And I am going through interphase, but this time I am a cell in the testes, in the seminiferous tubules. Okay, seminiferous tubules. The seminiferous, hang on one sec. Sorry, I have a hawk that visits my bird feeder, and I thought he just came in. Not that I want to see him wipe out any of my birds, but still, it is rather fascinating. Okay.
Is your bird feeder big enough to?
What?
Is your bird feeder big enough for like a hawk to just go
No,
in?
he comes and he sits on my porch, and he goes after the birds.
Oh,
So yeah, oh yeah. I've seen him, I think 3 times. One time I got a picture of him just sitting there. It was a fluke. But there are birds flying around now and they disappear for like 2 days if he comes by. So it couldn't have been him. All right, sorry about that. I get all excited about this. And I'm in my kitchen where I can see it. All right, so I have I'm in the seminiferous tubules. That's where sperm are made. And so I'm a cell in the seminiferous tubules, and I want to make a sperm cell. Okay, I'm actually in this process. I'm going to make 4 of them. So I'm getting ready to make my sperm cell, and I am in interphase. So in interphase, I'm going through gap 1, and then I'm going through S phase. And so in S phase, I am going to replicate my chromosomes here, see if I can get them all together. So none of that has changed. Gap 2 hasn't changed. So interphase this time is all the same. So we're in meiosis 1, but now I'm going to start mitosis. And here's where it gets a little hinky. First of all, what happens in prophase? Now this is prophase 1 because it's in meiosis, but what happens in prophase? Come on, I know you know because you can cheat and read it off here. What happens in prophase?
The chromosomes condense.
Chromosomes condense, and there they have condensed.
Then the nucleus
Can't read
membrane.
my writing is that's the nuclear membrane
I actually have a question.
is disintegrating. Yep.
Earlier we were talking about the different steps, the S1, S2. Then we drew a bunch of lines during mitosis. Is the metaphase and anaphase and all of that part of the...
Yes, yes. So on the sheet that you had, or the page way back, what page was it? 56 maybe? Oh, it's in there. It shows it there. So if you look at the top of 56, you'll see that after G2, you see prophase, and they make a prometaphase and metaphase and anaphase and telophase and cytokinesis. You see that there, Katie?
On page 56?
Yeah. Is that what you were asking?
Yes, I think so.
Okay. Okay, so we've gone through gap 1, synthesis, where we've replicated our chromosomes, gap 2, and now we're starting prophase, and prophase, our chromosomes have condensed, our nuclear membrane is breaking up, and what's happening? My centrioles are going to the poles, right? That's going to happen in any prophase. So you got to remember those 3 things. But when we get to meiosis 1, we have 2 other things that happen. Homologous chromosomes line up. So they pair up together. So this is my chromosome 4, and this is my chromosome 22, or whatever it was, right? I can't remember, but here's my chromosome 4, here's my chromosome 22. Both of them, both chromosome 4s line up together. Both chromosome 22s line up together, as do all the rest of my chromosomes. Now what happens is something, so that's the first thing that's going to happen is my homologous chromosomes are going to pair. And they're going to form something called a tetrad, or they're going to synapse together, but don't worry about that. Then they're going to undergo something called crossing over. Crossing over is going to increase the genetic variability. So what's happening, and I'll show you a little bit more closely here, is this leg crosses over with this leg. And a part breaks off, and an equal part breaks off the other side. And they trade information. They're trading that information. Okay? So I still have my sister chromatids, but now I've got some variability built in here. That's one way we get variability. And so all of our chromosomes can do that. All right, so my homologous pairs have lined up together and they have traded information called crossing over. Now what's going to happen? What's my next phase going to be? What's my next phase called?
Metaphase 1.
Metaphase 1. So remember now I don't have a nucleus or a nuclear membrane, right? But I do have my centrioles and my spindle forming. And in metaphase 1, I'm still dealing with homologous pairs. So my homologous pairs, whoops, gotta get them in the right pairs here. My homologous pairs are going to line up at the center in metaphase 1. Homologous pairs. Looks a little different from last time, doesn't it? And when they've lined up at the center, they're grabbing, right? They've got the microtubules, the spindle is grabbing the centromeres again. And this is going to move this way. And this is going to move this way. But stop. You can see right now which ones are moving which direction. So if we were to carry this on, these would move this way, these would move this way. But what happens, we're going to back up the taxi here. What happens if when we line up, since this is random, which one lines up closer to the South Pole and which one lines up closer to the North Pole? Which one is grabbed by a spindle from the North Pole versus which one is grabbed by the spindle from the South Pole? So we can have any combination in our homologous pairs for that. That is random alignment, or the other word is independent assortment. So when they line up at metaphase, it's arbitrary which one is going to line up and be dragged north and which ones are going to be dragged south. Okay, so that is independent assortment or random alignment. And what's our next? We're after metaphase. These two start pulling toward the pole. What's that called now? What comes after metaphase 1?
Anaphase.
anaphase 1, right? In this case, it's anaphase 1. So these are going to start pulling. And guess what's going to happen when they get up to the pole? You know this because we wrote it down. What's going to happen when they get up to the pole? You can cheat. What's going to happen to our chromosomes? They're going to decondense. We're going to start forming our new nuclear membrane. And cleavage furrow is going to form, right? Because those always happen, don't they? Those are always going to happen in telophase. All right. So that was meiosis 1. And we've got cytokinesis. So now, after meiosis 1, our parent cell has divided into 2 cells. Okay. Now what I'm going to do, let's see how best to do this. I'm going to, hold on, I'm going to make a second cell with this. That out of the way. That underneath there. Now I have 2 cells after meiosis 1. So here are my 2 cells. And where am I located again? Where's my cell located? Wasn't it in the testicle and the seminiferous tubules? Because I'm making sperm. All right. So now I have 2 cells that I'm working with, but the exact same thing is happening in both of them. Now, here's the thing. We need to make 2 more cells. So we still need new organelles. We still need new proteins, but we do not want more DNA, right? Because how can we get down to half the amount of DNA if we keep making more DNA? So between, now you can't see it, between meiosis 1 and meiosis 2, there is no duplication or I should put replication. I know you can't see that very well. Replication of DNA. Okay, so no replication of DNA between these. All right. So since there's no replication of DNA, I'm stuck here with what I've got. Let me, starting to get sick from this darn Sharpie. I like it better, but it's really stinky. Okay. So I get through gap 1, I don't make any new DNA. I get through gap 2, and now I'm at meiosis 2. So what happens in prophase? Always. Come on, we know this now. What's happening in prophase? Nate, what's happening in prophase?
The chromosomes will condense and the centrioles migrate to the poles.
Perfect. All right. And what's the one other thing that's going to happen at the same time, Darian? Do you remember which one he missed?
Sorry, I need you to repeat the question.
What is happening in prophase 1 or prophase 2? Sorry, prophase 2, but it happens in any prophase.
Nuclear
Chromosomes?
membrane.
What?
Nuclear
ahead.
membrane starts to fragment or break up.
Perfect. So between you two, we have that the nuclear membrane is breaking up, that the chromosomes have condensed, and that the centrioles are migrating. So both of our cells are doing that right now. All right, so let me see if I can reuse any of my papers here. Okay, so what happens in metaphase? What always happens in metaphase? Metaphase gets pretty boring because what happens in metaphase? What are our chromosomes doing in metaphase? Somebody's got to know this. Brian, what if a line in the middle? Thank you, Tyler. All right, so metaphase, my chromosomes in the form of sister chromatids, right? Because I've got 2 of them attached together, are lining up at the center. So Brian, what's going to happen in anaphase?
The sister chromatids separate and become daughter chromosomes.
Perfect. So during anaphase, remember that there's going to be our fibers, our spindle fibers attached to each center. Whoops, sorry, I'm showing you one you can't even see. Attached to each centromere, and they're going to start pulling away. So all my chromosomes, my sister chromatids, are going to separate. Awesome. We're almost there. All right. So then they keep moving, right? Anaphase, they keep moving. They get up here. And what is going to happen next? We've gone through anaphase. What's our next phase? Brian, what's our next phase called?
Telophase.
Telophase.
Telophase 2, but yeah.
That's right. Perfect. Thank you for reminding me of that because I would have gone right by that. So we're in telophase 2. So what am I doing here in telophase 2? What's happened to my chromosomes?
They've recondensed.
They decondensed, right? So they decondensed And I now need a nucleus for my cell. So I'm making a nucleus. Whoops, don't have my nucleus yet over here, do I? And what else is going to happen?
The cleavage.
The cleavage furrow is going to form. So I'm going to start seeing my cleavage furrow. And what is my last step going to be? What's that called? That last step that's going to end up with cells looking like, without the butt crack on the bottom, but the looking like the glasses there, the donuts. What's that called? Cytokinesis, right? Okay, so now I have 4 cells. Use your imagination here. These are going to be 2 different cells like this. I now have 4 cells at the end of meiosis 2. Because each of those cells that I had at the end of meiosis 1 has now divided. But I want to, just to make this clear, let's look at these chromosomes that we have left here. Okay, so I've decondensed, but I'm going to, for purposes of showing what our cells look like, we have 4 different sperm cells. And notice that this one, this is dad's chromosome, what was it, 4, and this is mom's chromosome 22 in one sperm cell. This is mom's chromosome 22, but notice what's happened to dad's chromosome 4. It now is quite a combination. Here we have dad's chromosome 22 and mom's chromosome 4, and she's got a mixture of mom and dad on that chromosome. And then, of course, we have this one that has dad's chromosome 22 and mom's chromosome 4. All 4 of those sperm, and we're only talking about 2 chromosomes here, aren't we? We haven't got a whole collection of 23 chromosomes. And we can see the variability in each of those 4 cells. So now our last step is this is the sperm that's going to combine with the egg. This is the one that's going to make the journey, and it's going to swim all the way up there, and it's going to show its prowess, and it is going to fertilize the egg. But the next one in line was this one. And another one, we had a good batch here, right? Because there are billions of sperm aiming at that egg. So whatever egg happens to be the one that combines with the sperm is going to add more variability, isn't it? So remember, the egg is doing this as well in a little more limited fashion. And we'll look at that in a second. But that is our last mode of variability. And that's called, what do we call it? Fertilization. It occurs during fertilization. It's random fusion of gametes. Let's see if that's actually on here somewhere. No, it's actually not. Not where I thought it was. But I think we get to it, so. All right, so there is our, that is our mitosis and meiosis, all right? And that's how, when we get to the end of this in meiosis, how we get the random fusion of gametes, okay? So are there any questions about mitosis? Well, let's now skip mitosis right now. Any questions about meiosis? Okay, so if there are no questions, the 62 and 63 are good references. Page 64, you can use if you want to practice. But now we're going to the bottom of page 64. And this is about meiosis 1. We're only talking about meiosis 1. So do the chromosomes inherited from the mother or the father, we did red and yellow, have to remain together during meiosis 1, or following meiosis 1, sorry, following meiosis 1, did they have to stay together?
No.
No, they didn't. Basically, the same genetic material does not stay together. And that occurs because of the crossing over and the random alignment or independent assortment. And they're calling in between our interphase between meiosis 1 and meiosis 2. Notice on the top of page 65 that they call that interkinesis. I don't use that term, but just so you know what that means. And the important part in what they're calling interkinesis is that our chromosomes don't duplicate, right? We do not want our chromosomes to duplicate there. And then the bottom of page 65, first of all, before we go there, does meiosis 2 look more like mitosis or meiosis 1? If you think about what we did here, does meiosis 2 look more like mitosis or meiosis 1? Any ideas? Think about meiosis 1, and we had homologous chromosomes that paired up. We had crossing over. During metaphase, we had homologous chromosomes lined up at the middle. So meiosis 1 and meiosis 2 are not very similar, are they? So meiosis 2 is very similar to mitosis. Okay, keep that in mind. Because what's separating and what's lining up at the center in both mitosis and meiosis 2 are sister chromatids, right? Remember, the sister chromatids line up in both of those. The homologous chromosomes are only found in meiosis 1. Hint for the quiz, because I always ask something about homologous chromosomes. And if I'm talking about the homologous chromosomes doing anything, it has to be in meiosis 1. Everybody got that written down? That will never lead you astray. That's always a good hint. Okay.
So, sorry.
Go ahead.
So meiosis one, everything that, I mean, what was the word, the other M word you used? Metaphase?
Tell me what it was in reference to. Metaphase is in. Oh, are you talking about the homologous pairs lining up at the center? That is metaphase, right? That's happening in metaphase 1.
Okay. And I don't, half of this stuff I'm trying to figure out how to spell.
I don't care on your paper what it looks like. So, okay. So let's look at the bottom of page 65. And this is going to give us a summary. Oh, there we go. That's where I was looking for that. Okay, number 1, the parent cell has the diploid number of chromosomes, and the daughter cells following meiosis have the blank number of chromosomes. Remember, we're talking about sperm now. Half, right, the haploid number. And that means meiosis is cell division in which the chromosome number halves. Number 2. And number 3, if a parent cell has 16 chromosomes, the daughter cell has to have how many chromosomes after meiosis? Right, 8. Right, Megan? 8? Tyler beat you to it. Whereas meiosis reduces the number of chromosomes or chromosome number, fertilization restores the chromosome number. A zygote contains the same number of chromosomes as the parent, but are these exactly the same chromosomes? Are they the same chromosomes, Nate?
No.
No, they're not. They've got some genetic variability in there. And what is another way for sexual reproduction, a way that sexual reproduction results in genetic recombination? Brian, what was the term I used? Random?
Hold on, now I gotta go back. I wrote it down.
What's
but I can't
happening?
remember it.
What's happening? You tell me what happens and I'll give you the word.
Random alignment?
Random alignment is going to happen in meiosis 1. What happens when the sperm and the egg get together?
Fertilization?
Yep. But it could be this one or this one. What was that called? Anybody remember? Oh, do you remember, Megan?
Yeah, the random fusion of gametes.
Random fusion of gametes. Random fusion of gametes. Awesome. So we have 3 ways that we get genetic variability: crossing over during prophase 1, random alignment, which is also called independent assortment in metaphase 1, and then random fusion of gametes during fertilization. Okay, so those are the 3 ways that we get variation. All right, so let's look now at page 66. And let's look down the differences between mitosis and meiosis. We should be good at these now. The number of cell divisions in mitosis is how many cell divisions do we have in mitosis?
1 turning into 2?
Yep, we have one cell division, right? Because that's the cytokinesis, and our one cell, you're right, turns into two. So we have one cell division. How many do we have in meiosis?
Would it be two of them?
It is two of them, and that's how we get four cells. Okay, so our chromosome number in our daughter cells during mitosis, is it going to be the same or different than parents? Or parent cell? Is our chromosome number going to stay the same in mitosis?
Yes.
Yes, it is. Yes, it is. And yet in meiosis, sorry, I have a cat crawling up my leg here. In meiosis, it is going to be half the number of parents, right? Of the parent cell. And in mitosis, how many daughter cells do we have? Mitosis, we have two daughter cells, right? Versus how many in meiosis?
4.
Four cells.
What's your cat's name?
Oh, this is Zeke.
Oh,
Poor, I hesitate to put him in front of you. You're going to wonder why I still have him. Poor little guy. He's skinny as a rail. He got really sick a couple weeks ago, but he's getting better. But, well, you can't see him because he's a black cat, and I'm holding him down, but he's skinny. Anyway, so mitosis compared with meiosis on the next page, on page 67. So under mitosis, it says in prophase that we don't pair the chromosomes, right? But what do we do under meiosis 1? We pair the homologous chromosomes. The other word for that is homologs. What happens at metaphase? Metaphase, our sister chromatids line up at the equator. What happens in meiosis 1? Or sorry, metaphase 1. Well, yeah, there we go, metaphase 1. Our homologs line up, right? Don't our homologs line up at the equator? And whereas mitosis in anaphase, my sister chromatids separate. In anaphase 1, my homologous chromosomes separate. Right? My homologs, my homologous chromosomes are going to separate in anaphase 1. And then in telophase, in mitosis, chromosomes have one chromatid. But in meiosis 1, there is two chromatids, right? So the chromosomes have two chromatids. Anybody have any questions about that? You can actually, I think it's on the, yeah, they compare mitosis 1 and meiosis. And you can see that on the bottom if you compare the two in the pictures. And on page 68, mitosis compared to meiosis 2, they're actually very similar. Prophase, there's no pairing up of chromosomes, so the homologous chromosomes don't get together. But in metaphase, the sister chromatids, they're calling them duplicated chromosomes. But the sister chromatids are going to line up in the center for both metaphase and metaphase 2. Sister chromatids are going to separate in both anaphase and anaphase 2. And in telophase and telophase 2, we're going to end up with cells, but there's where the cell number is going to be different. Telophase of mitosis is going to have 2 diploid cells. Telophase 2 is going to have 4 haploid cells. You need me to go over any more of that and read it again? No, I went kind of quickly. Ah, now my nose is itching. Okay. The next, no, stay down. The next stage is gametogenesis. And gametogenesis is what we call making the sperm and the egg. Okay, so we practiced here like we were doing spermatogenesis. And you can see up at the top, there's pictures of how those 4 cells are made on the right-hand side. And when we call making of sperm spermatogenesis, we call the making of an egg oogenesis or oogenesis. Either way is fine. And so let's look at 70, page 70. And I'm just going to give you these. You actually, I think, probably know all of these. But gametogenesis is the formation of gametes. And we know that human gametes have a haploid number of cells. So we know that they have 23, or not cells, I'm sorry, chromosomes. So they have 23 chromosomes. Spermatogenesis is formation of sperm. And oogenesis is formation of the egg. Both of those are going to have 23 chromosomes in a human. And following fertilization, we will restore that full number, the diploid number of 46. I want to show you one more thing, so don't get all excited yet. But while I'm on this page, page 71, I think you can do that entire page. Okay, so page 71. And then the other assignment, both of these are due on Sunday night. The other assignment is to do the virtual microscope and take pictures of each of the cells on there, each of the phases. And the directions are in the assignment, so they're a little more specific. The other option is if you have been writing them, if you have pieces of paper where you have those phases on them, you're welcome to take pictures of those and not do the virtual microscope. Okay, so I'll take either your pictures or the pictures from the virtual microscope. But you need to have all of the phases that are required. Okay, but I will do either of those. Now, last thing I want to show you, and we'll talk about this a little more later in the semester, but while I've got these beads, I want to talk about something called non-disjunction. So actually, while we're here are our 4 sperm cells, right? And each one of these has 2 chromosomes in it, right? Each one has 2 chromosomes. So let's figure out how many chromosomes we started with, and we can count backwards for this. So each one has 2 chromosomes, all right? But these whoops, I played with them. Should not have played with them. That goes here and that goes here. Oh. There we go. All right. Not the way I really wanted them to be, but nonetheless. Okay, so we're gonna ignore these two sperm here, and we're just gonna play with these sperm for a second, and I'm going backwards now. So these are coming back together in the center, right? Well, I'm gonna play with these two because it won't work otherwise, and these will come back in the center. Now, I'm gonna go back to the cells before this. So this is what I started with. Now it's going to be easier to do it this way. Sorry about that. But this is the cell I started with. So how many chromosomes did I have in that cell? I ended with 2, but these are all the chromosomes that I started with. I started out with 4 chromosomes. So that's where we can see that I got the haploid number from the diploid number. Now, what I want to show you was, let me see if I can find I'll use this as a little review. Okay, so we start out and I'm going to be in meiosis 1. And remember that in meiosis 1, I have, first of all, prophase. My chromosomes are condensing. My nuclear membrane is going to disintegrate. My centrioles are moving. And my homologs, my homologous chromosomes are going to line up. So my 2 number 4s are going to line up, and my 2 number 22s are going to line up. And then they're going to exchange information, right? So crossing over has occurred. You can see that here. Then they're going to line up in the center during metaphase, and that's random alignment. But now watch what can happen. Whoop, hang on, let me find. Okay. So they're lined up at the center, and my spindle's holding on to this, and my spindle's holding on to this. But what happens if my spindle either, let's say this separates and this goes this way, and this one separates and goes this way, but this goes this way. There's a mess up here. And all of my chromosome 22s go into this cell. And so this cell now lacks a chromosome 22. That's called non-disjunction, and we'll talk about that in lecture. But non-disjunction accounts for things like trisomy 21, which is Down syndrome. Having an extra chromosome is caused by non-disjunction, meaning that they don't separate. Another way we can see this, okay, so let's go back here. That was meiosis 1, and all of our chromosomes went one direction. But what happens if during meiosis 1, everything works fine? And then we've got these 2 cells that are going to separate. So I'll move that out of the way, but this one goes to separate, right? We have prophase, and then we get to metaphase, and it lines up. Our sister chromatids are lined up. Our spindle grabs, but there's an error somehow. And this one separates and goes that way. This one moves this way, but both of these come this way. Okay, and in the case of a completely missing chromosome, so if this was the sperm that ended up fertilizing the egg, that is not going to be compatible with life. So that zygote will not survive. This, depending on the extra chromosome, that can obviously, you know, Down syndrome, and that is compatible with life, but you know that also comes with disabilities. So non-disjunction can occur in both meiosis, or either, I should say, meiosis 1 or meiosis 2. And that's how you see diseases that, or conditions that are related to the loss of a chromosome or the gain of a chromosome. You won't see the loss very often. That is relatively unusual, but you do see several diseases or conditions that are associated with the gain of a chromosome. Okay, that's called non-disjunction. So just while we're there, while we got all that stuff out, I wanted to show you that. And that does it for me. Unless you have questions or you want to hang out with me to do the problems, you are free to go and have a great day. I hope you get to enjoy a little bit of the sunshine. Looks like Megan's house is sunshiny. Oh, same with Katie. I can see the sun behind you guys.
Yeah, Dexter's it's pretty sunny right now.
Well, get out and enjoy it. Or hang out with me, one or the other. Hello, whoever you are.
It's my roommate, Chandler.
Hi.
I wanted to feel special. Anyway, I had a question. So you said that we could draw the steps of mitosis and meiosis instead of using the microphone and sending those pictures in?
Mhm.
Okay. Sounds good. Thank you.
You're welcome. You all set, Taylor?
Well, hope you enjoy your day. Hopefully it's
Thank
sunny.
you. You too, Megan. I'll see you tomorrow.
See you tomorrow.
60 degrees tomorrow. Yes. All right.